back in 2002. I did a PhD in New Mexico, where I worked on semi-arid grasslands and ephemeral streams. So I've always been interested in water and the terrestrial aquatic interface and the microbes that live there. How do they do that? How do they affect the chemistry of the soil around them? And then I did a postdoc in Oregon and another one in Alaska, focusing on nitrification and denitrification, so soil fertility, which is driven by microbes, and drought tolerance of microbes here at Konza, in fact. And the third Alaska topic was about the recovery of soil microbes and organic matter after a volcano erupted in Alaska. So it's all, I have wide interests. That's why I like writing a long overview of microbes of the prairie region. And I still have quite a bit of research going on around the world, but mostly focused here in Kansas now. So I'll go through this real quick. This is my spiel for everybody. Microbes, as you know, after reading that fact sheet, are abundant, diverse, ubiquitous, and small, but they come in a lot of different forms, more like they have a lot of different shapes and sizes themselves, which are related to their different functions, their different jobs in the environment, and how they tolerate changes in the environment. These are just some pictures to illustrate the point. And lab, since I was working there, we actually collect soil or water from an explicitly understood field context. So Konza is amazing for studying microbial ecology because we know the history of the landscape and the land management here, and so much about plant diversity and soil chemistry. So we have this strong field context. We bring soil back to the lab, extract DNA and RNA, and cells, carbon, nitrogen cells, measure those things to tell us something about this one is the below ground plots, the fungal community. So I can come back to these later if you really want to know, but this just says the fungal community in the soil changes with woody encroachment and fertilization. So we figured that out by extracting the DNA and making this graph. That's one of the fun facts that we'll come back to later. I'm showing you this, I should explain it. The circles are the annually burned soils, and the squares are the unburned, which, as you know, like right behind us, the unburned plots are covered in trees. And they fall in a different place on this graph, which means they have a different type, relative abundance and different types of fungi. So we can piece that information out a little more. This is an experiment that we ran in the lab on soil from the ramps, the rainfall manipulation plots, which are also back over there. So that ramps experiment, when it was operational, half of those plots got ambient rain, like they collected the rain into those tubs and then they didn't sprinkle back the same amount of rain. The other half, they collected the water in those tanks, held it there for a while, put it back out. So it was the same amount of rainfall, but bigger events and drier intervals between each event, so it was more droughty. That affects the way microbes respond to a rainfall. That's what's shown here. So after, especially that altered, more droughty history, there's very few types of bacteria left in the soil that can survive a dry down event based on what we measured in the lab. They can just put soil in a jar and measure the response. So there's some local extirpation of resistant microbes after you subject them to drought for a while. And this last graph is an example of some river water, Kansas river water microbial communities that we filtered out onto these filters. So that's a bunch of algae and bacteria that were floating in the water. we inoculated these communities back into water in jars that either came from the river or came from the farmland fertilizer plant in Lawrence that dumped into the river. So we did this just recently. That's actually where these microbes came from. So this microbial community from the farmland fertilizer plant water has a very different response in its activity to nitrate availability than the microbes that live naturally in the reservoir, which is what we expected. But the microbes in the river water are more active at the concentrations of nitrogen in the river water. So we can tell the difference using these DNA and chemical assays. We can tell the difference in the community composition and activity of different communities from different histories. We can start to think about how that might affect water quality or soil chemistry in the future or in different situations. So that was a lot of graphs. That's the intro I give to the graduate students actually. But that's more like what I do during my workday, think about problems like that as well as teach. I have students, they're sitting out there by the ramps collecting soil. This is our field laboratory. So we bring out some of our lab to the field and sieve soil wearing gloves so we don't contaminate the soil with our own microbiota, collect it, preserve it properly for DNA extraction, take it back to the lab and do more with it. That's my intro to myself and my research. And from there, I thought I would just dive into the document that you all have. Any questions for me right now? As we go through the document, we'll come back at the end to the fun facts about Konza Prairie microbes, which could include questions about some of the stuff I just showed you. And incidentally, Jill said you can contact her with questions. You can also contact me. And I actually don't mind if you share these slides too. I'm not sure what. Certainly my email address, you're welcome to write down your question and send it to me and I'll answer it. So what I'm trying to do now is put the through it, and I might jump back and forth as needed. Other slides. This is weird. Okay, first section. What is microbes? Latin prefix for small. That prefix attached to a lot of words. Okay, so that micro symbol modifies your measurement category to say micron, which is smaller than a millimeter. The next increment of tenths down from millimeter. So here we are. We're like a meter tall-ish in that range. The ants is more in the centimeter range, or 1,000 times smaller than us. 1,000 times smaller than that would be frog eggs. And 10 times smaller than that would be animal cells. 10 times smaller than that are archaea and bacteria. So we're thinking at orders of magnitude of smaller size in terms of how the organism interacts with its environment. In share on the internet. But this is an example of what the environment looks like to a bacterium or a fungus near a growing plant root. This sort of huge input of carbon is coming from the plant root in the soil, and then you see a lot of these little dots. Those are bacteria that tend to be clustered closer to the root where there's more energy, and then carbohydrates are going their way through the matrix there. So, any questions? I can ramble about this stuff a lot. I just don't want to be too boring. I'll continue. I have slides on the scale of bacterial cells to large cells. So there's this, I use this in my class. There's a figure that you just saw. Again, for reference, this microscopic image shows a human skin cell. There's the large outline there. That's its nucleus, this dark cluster where all the DNA is concentrated. And then all of those little dots are bacteria that are contaminating the skin cell surface. So again, just to put it all in perspective, bacteria are tiny. You probably heard that the number of bacteria in and on the human body outnumbers our own cells by quite a bit. Those estimates have been downgraded recently because bacteria are not distributed evenly throughout our entire body. They're concentrated in our guts and on our skin. So the cell numbers might be more in the range of 1 to 1 or 1 to 10 bacteria, 1 to 100. There's still a lot of them relative to our own cell mass. But they work for us. In our guts and on our skin and out in the soil, they also work, they can work for us.
Question?
Yes.
You go out on the prairie and dig down from the surface of the soil. Would you find a gradation?
Yes.
At what point in a soil like the prairie soil that has not been worked and turned over and like that, would you get to a point where you no longer find any microbes in your plants?
So the question is, when you move down from the surface of the soil to depth, is there a decrease in the number of microbes? Yes, definitely. And the second part of the question is, when do we find a point of no life, no microbial life? And that is harder to answer. There are known communities and populations of bacteria at hundreds of meters below the surface that have been extracted from gold mines in Africa, for example. There are bacteria that can live off of rocks, get energy from the chemicals in that are dissolved from the minerals in rocks. So the limit to microbial life at depth could be related more to pressure and heat that doesn't allow a cell to remain intact anymore. But we have very little information from the depths of the earth about the microbial life.
Does that mean they drop off? Or you skip this day after you get all the roots on?
Yes. Yeah, so the distribution of microbes in general, like all life, is related to the availability of energy and carbon. In certain soils where there's a lot of carbon and energy coming in from plants, that's the highest ones.
I guess the basic question is, since they can't move, how did they get there?
Water percolates down and through. There's one hypothesis out there. I know some folks in England are studying this, and I'm not sure. There's so many areas of microbial ecology that I'm not an expert in. I'll tell you little things that I do know. Because the Earth's geology is driven by the movement of molten rock, and like there are connections in groundwater and liquid moving vectors of materials at really, really deep levels and really distant locations. There could be connectivity in the subsurface for microbes to move around in, like continentally. That's not proven, but somebody's thinking that way. In the global oceans, ocean currents move constantly. There's a lot more mixing of microbes locally. So there are cosmopolitan, widely distributed open ocean microbial types that we know are found everywhere. They move with water. Yeah.
So it does not have to be water that's filled with microbes. It could be anything that's filled with like oil.
Yes. Could be plant roots themselves. It could be infrastructure that humans have put in. It could be fossil fuel deposits. There are some microbes that extract energy from those petrochemicals. There's obviously a lot of energy stored there. You see them bloom like in the Gulf oil spill. There was a bloom of microbes that can live on oil out there after the Deepwater Horizon oil spill. So cattle moving around, bison moving around. That was in my fact sheet. I alluded to the fact that just animals moving around the landscape also carry microbes with them. So even though these cells are small, some of them can swim, some of them can't. They're not necessarily, their ranges are not necessarily tied to where they can move under their own energy. Yeah, please interrupt me. And I'm going to try to repeat your question in case you want to be able to hear it later.
So I think one of the underlying questions is what makes a prairie a prairie? And the microbial composition of the soil is one of the defining aspects of a prairie. And one of the best ways to get rid of the microbial life of a prairie is to plow that said prairie.
Mm-hmm.
So then the next question would be, how do you get the microbial population back? How do you get it back, and can you get it back?
Let's go.
In my mind, that's the heart of the question.
We'll start talking about carbon and decomposition. A lot of microbial ecology is tied to nutrient cycles and the loss of the energy in the habitat and the organic matter that is driven by tilling of soils. Also dramatically affects the microbes that can live there. So I'll come around to your question, but I'll kind of start talking about carbon first. Most microbes, most bacteria and fungi use carbon to grow and get energy the same way we do. They take in sugars, assemble those sugars into new biomolecules, or breathe, respire those sugars using oxygen and release carbon dioxide. There are some really cool microbes that don't do that, but most of them are supported by organic matter, just like us. Over time, not just decades or centuries, millennia of soil development, with concurrent with plant growth in particular, a large amount of organic matter from those plants accumulates in the soil. You talked about, you put in mollisols here. That's our classic grassland soil type across the Great Plains. There are other soil types that have a fair bit of organic matter. Some soil types that have more organic matter than mollisols. But it has literally taken millions of years for organic matter to accumulate and for the associated microbial communities to develop. Tilling physically breaks up the soil aggregates that hold a lot of that carbon. So it's not, and the loss of carbon and the death of a lot of the microbes associated with that carbon is directly related. So the physical disruption of soil aggregates releases carbon that was held within those aggregate structures that wasn't available for microbial respiration for hundreds of thousands of years. The microbes, the bacteria and fungi that are there, it's very excited to have this new food source. You see this when we take the soil back to the lab and kind of break up the aggregates. There's a flush of respiration, which is driven by the activity of those cells. And all that CO2 goes back up into the atmosphere. And in order to get it back, we need more plants and algae to fix that carbon and put it back into the soil. So the biggest question about can we get back the microbial community, the biggest factor underlying the recovery of the microbial community is the recovery of that complex organic matter habitat, and aggregate structure, and complexity of the types of carbon that are in the soil for the microbes to live on. So soil prairie restoration experiments that have tracked microbial community development see a very strong, including work from Sarah Baer and some newer work from Nashua grasslands over in Illinois. Done a good job tracking the microbial recovery. It follows the organic matter accumulation pretty closely, which makes sense. I see that too in the volcano work. There's a really tight link between the few plants that have established, put new sugars into the soil. You see the diversity and composition of tens of thousands of different microbes come back within a couple of years when that particular type of organic matter is available, like the native type of plant-derived organic matter. Out there is a different type of grass, and it's a different microbial community. But when that grass is present, putting more organic matter into the bare ash, you get surprisingly quick recovery of that pre-disturbance. So there's a pretty close link. What makes a prairie microbial community a prairie microbial community? That is, I can tell you a lot of, I can tell you certain things we measure that are characteristic of prairies. And then there are other things, other aspects of the microbial community which are more indicative of the soil type or the pH or alpha salt versus low salt. You have grasslands on alpha salt. All you need for a grassland, which is like a classic forest soil, or an erythritol, which is a classic desert soil. The main, like a really forest level, what maintains a grassland? The disturbance, the fire or the grazing or the combination of factors that allow grasses to remain dominant is not necessarily tied to the soil type. So South Africa, really, really old soil, very different soil type, very different microbial communities, classic grasslands because they still have fire grazing. So as far as measuring the, more interesting slide up here, measuring the community composition, I can't necessarily tell you that there's grass above the soil, just from this snapshot of like what types of bacteria live there. Even back there on the below ground plots, the bacteria did not care whether there were trees or grass growing. They care a lot if fertilizer has been added. They've changed a lot of the amount of fertilizer added. And they've changed more under the trees back there with the same amount of fertilizer added. But the bacteria themselves don't care whether there's trees or not. The fungi, which have evolved with plants, and have species that are more associated with certain types of plants, or maybe better adapted to decompose certain types of plant litter. The fungi do care that there are trees there. I can show you graphs on that. I don't know if that would help or not, but I do have a slide on that one. Yeah.
Here on Konza, as well as other native prairies where people are trying to restore them, there's a big emphasis on getting rid of invaders. And often herbicides are used and various other chemicals, as well as mechanical removal of these plants. How does that affect the microbial community of the prairie?
It can, in maybe in surprising ways. That's not, so the question is, how do pesticides or herbicides affect soil microbial communities? In indirect ways, the plant dies, it could feed the decomposers in the soil. The microbes might not respond directly to the pesticide or the herbicide. Some microbes actually degrade those particular chemicals. Roundup in particular has a lot of phosphorus in it and could actually, once it degrades, be a new nutrient source for some microbes. So I don't worry about the pesticides and the herbicides affecting the microbes too much, especially at the rate that we're pretty careful about how much you
I
have.
think it depends on the chemical used.
Yeah.
If it's got chlorine in it, then that might be.
Also leach through. Yeah, so that's like the levels of which I can answer. Certain invasive plants themselves can release chemicals into the soil that have negative impacts on microbes that might be beneficial to the other plants around them. So the Eurasian bluestem that's also just right over the hill there, that and the restoration experiment that's going on back there, it is predicted that the Eurasian bluestem is allelopathic, it attracts certain microbes and repels other microbes that are beneficial for its growth alone and negative for the other grasses' growth. So they've killed off, using solarization, as much of that Eurasian bluestem as they could, and these are folks from Oklahoma State. They've planted some native plants and native seedlings in there to evaluate whether or not those native seedlings grow as well after the Eurasian bluestem has been in the soil. So I think the point is the invasive plants themselves can have as great an impact on the soil microbial community, potentially in a negative way for native prairie, as some of the removals. But I haven't seen, there might be something out there, I can't like directly compare good and bad levels of benefit or negative. There might be more complex.
I have a lot of experience with vacation bluestem. In my observation, I'm not a scientist like you are, it is allelopathic to itself.
Okay.
And if you just leave it alone and watch it, it's very clumpy and eroded between the clumps.
Yeah.
I've had a ranch that I don't own that I use for almost 100%. And we stock it at about 7 times the stocking rate. We keep it about this high. And over 4 years, that has really thickened that up. It's still in between, but before it was really spread. And I'm curious. I guess I'm throwing that out as something to research as what's going on there.
Yeah, over time there can be further feedbacks of one dominant plant being established. Even the dominant big bluestem up there, that has potential feedbacks. But specifically, I mean, I don't know much more about Caucasian bluestem,
Other
I
than
just told you, so.
If the foot is stressed, come back and reduce it and decided to,
Hmm.
for some reason, I'm not sure there's a reason.
That's- A lot of people. I don't know either. Yeah, so there's so many, there's a lot of questions I can't answer about. Maybe one expert could, and maybe we need to do more research.
And I have one other question. You said that it's different between forest or trees and prairie.
Mhm.
Okay, and I'm gonna ask you, is there a difference between invasives like
Fungi.
brushwood, dogwood, and forests? Is there?
I'll tell you something.
Did you repeat the question? Excuse me, did you
Yes.
repeat the question?
The
I
earlier
couldn't hear.
question I couldn't repeat, it was about Caucasian bluestem establishing and having feedbacks that prevented its expansion, even with grazing, and I couldn't answer the question. This question is, with different types of woody encroachment, do you also see different types of microbes underneath the different species of plant? I have a student this summer who's going to evaluate that question very directly. I have data from the soil itself, but not the plant root specifically. So that's something that we're really interested in too. There's a greater probability that the plant species composition, the plant species composition is correlated to the fungal species composition back there. So each of those plots has a different, two of them have cedar, and one of them is all brambles and sumac and stuff, and another one is hackberry. So they do have different fungal communities. We haven't gone in and looked at the specific associates with each of the big invasives. We're going to do that now because we're interested in that question. Something unique about the soil fungi under woody encroached prairie, like right here where I've studied it, the soil, different types of fungi that live under those trees are not the same as what you would find, really coarsely, what you would find in forests in Wisconsin or in Oregon or in the northeast U.S., which are native hardwoods. Those are forest types that other researchers have spent a lot of time understanding and know a fair bit about what the different species of fungi mean for organic matter degradation and such. Even though woody encroachment here changes the fungal community, it doesn't change it to something that we know about. And that has implications for nitrogen cycling and organic matter decomposition. So it's actually a pretty big area of further study. And a big surprise, really. Grasslands microbes under grasslands don't necessarily look like microbes under forests. And this is kind of a new woody encroached prairie, an even different type of microbe. There's probably the history of the prairie includes positive feedbacks between grasses growing and their fungal symbionts, which tend to be arbuscular mycorrhizal fungi, which is a completely different order and a completely different type of relationship than the ectomycorrhizal fungi that a lot of other trees associate with. So all of the trees that we find out here,
Microbial fungi.
okay, the trees that we find out here don't associate with ectomycorrhizae, they associate with arbuscular mycorrhizae, like grasses. So they're, sort of pre-adapted or better able to associate with the types of fungi that we already have. I think I have a slide.
So, let me see if I understand you right. You're saying that the bacteria can be much more of a generalist, but the fungi are going to be specialists to their particular plants that are found in the region.
More likely to. Because fungi have evolved with plants directly or indirectly.
So if I were trying to restore a prairie, could I then find some feeder, some fungi that I could purchase to introduce to my soil to help turn my prairie back into a prairie?
Possibly, you could buy some, Jim Beaver's group over at KU has a mixture of arbuscular fungal spores that might help. It probably wouldn't hurt, it wouldn't be guaranteed to help.
But it's the fungus, it's not the bacteria.
Yes. Okay. This is a slide showing you the rhizosphere, the area around the plant root where there's a lot of carbon coming out of the plant root and there's a lot of bacteria living. It's also an area where the fungal symbionts that have colonized plant roots are foraging for more phosphorus and more nitrogen. So this is a schematic of an arbuscular mycorrhizal fungal colonization, not infection of a plant root. It lives in between the cells of the plant root, and carbon from the plant root moves into the fungal cells, feeds the fungus, and then the fungus can forage further than the plant root can and bring in more water and more phosphorus and send it back into the plant root. Arbuscular fungi like that literally colonize inside of the plant tissue. They're a completely separate fungal group from the ectomycorrhizal fungi, which, as the name suggests, only live on the outside of the root surface. They form like a sheath around the root itself, but they don't get right in between the cells. They also range further, the ectomycorrhizal fungi. The trade-off is they can move further out into the soil and create these hyphal networks that are record size, acres of fungi in the Pacific Northwest. Those are all ectomycorrhizal fungi that have a larger foraging range but don't interact with the plant root as directly as the arbuscular fungi. So their life histories are very different, and we don't find as many of them here, especially in association with plants. And that has big implications for how plants
So we
interact
don't
with
have
the soil.
ectomycorrhizal fungi here.
The trees that we looked at right behind us, none of them are known to associate with ectomycorrhizal fungi. We will look at that more this year. But it's
Mycorrhizal
something
fungi grow on grass roots.
It's rare. Most grasses are arbuscular substrates. There are a few plants. Jill asked if there are ectomycorrhizal fungi that associate with grasses, and the short answer is no. Most grasses associate with arbuscular fungi. And then I was going to add there are some forbs that don't have any mycorrhizal associates, like garlic mustard. There's some others. There are very few plants that don't have fungi helping them bring in nutrients from the soil. I'll let him ask you that question.
So I'm wondering if you've looked at like a restoration site, and as this restoration matures over time, assuming that it is, do you see large changes in the mycorrhizal fungi involved or the bacteria involved?
Yeah, the question is, over time in a restoration site, are there large changes in the mycorrhizal fungi or the bacteria? Yes. So especially when the restoration site is from an ag field that has lost a lot of organic matter and a lot of microbial diversity. Again, I'm thinking of this Nashua forest, or Nashua grassland. You can see the graph in my head. They tracked, I think 20 years, like 15, 20 years of microbial recovery with some native reference site there. It was after a decade, 12, 14, 15 years, when the microbial community started looking more like the native site. And it changed dramatically, even in the first few years of restoration.
Kind of continuing on with that, have they found specific grasses or forbs that seem to increase the
Are there specific plant types, plant species, grasses, or forbs that increase the microbiome? Like most, I can't say definitively. I can say that the more different types of plants, the more likely you have a more diverse microbiome as well. And I can also say that a lot of the, I've already said, a lot of the recovery is related to the amount of organic matter coming into the soil. So plants that have greater root growth, and a greater impact on the soil itself will more rapidly promote the reestablishment of the microbial community as more diverse. So one plant or another, hard to say.
So in this symbiotic relationship, is a major benefit for the fungi having access to water?
Yeah, that's one of the two big things. The first benefit that we normally think of our vascular fungi is the phosphorus acquisition. Especially around here, this soil is pretty phosphorus limited for various reasons. Not all mollisols, not all grasslands are phosphorus limited, some are more nitrogen limited as related to the soil chemistry. But the vascular fungi are really good at releasing phosphorus from the soil minerals. But a secondary benefit that is established is more water acquisition. Just because the soil pore space is, especially the more aggregates you have, soil pore space is full of aggregates, and it's complex. It's just a straight thing. You've got large soil aggregates, you've got little pieces of sand, you've got little bits of clay, you've got, at the scale of the microbe, the micrometer scale, a really heterogeneous matrix for the cells to live in. Roots are bigger than fungi. The fungi can get into those tiny little pore spaces where the water sticks even after the soil looks dry. So fungi are better at getting water on a really dry soil. Soil can never be 100% dry. There's always, there's a point where the water potential drops so much that there's no connection within the matrix anymore between all of these little water films that are in these tiny little spaces. There's still some water in there. And the fungi are better at getting that.
General thing. What I'd like to know is, what is your goal? What are you seeking to research? Why? What do you hope the results will be? A sense of what drives you here in this area? What makes you decide to research this versus something else?
I care about soil fertility and water quality. I was a biogeochemist before I was a microbial ecologist. But the microbes are the organisms at that really small scale that are controlling the availability of different nutrients. So knowing which ones are better at denitrifying, like removing nitrogen from a polluted area at certain times, can tell us we use that, the one that we know about, to remediate another site? Can we use the sequence information that we collect to identify a community or a soil that would be better at withstanding a lot of nutrient stress than a site that isn't prepared microbially to remove a lot of nitrogen? There's so much information embedded in the DNA signatures that we get from the microbial community. It has to be useful in some way. So what bits of information indicate a soil that retains a lot of nutrients or allows a lot of nutrients to go away? That's like generally one of the things that motivates my work. Is what's the question?
Suppose you found microbes that really did everything you want. How would you propagate it?
You can train, this is something I tell them, how would you propagate the microbe that you have identified as beneficial? The way you manage the land affects the growth and propagation and thriving of the types of microbes that it's like gardening. You can tend the land in a way that promotes the growth of the types of microbes that you like. Wastewater treatment, but there's no one thing. No. And in fact, there's no one magic fertilizer or magic inoculum that you can put on your restoration to make it more like a prairie. You would need to also maintain the disturbance regime. And another thing that defines a prairie and underlies the microbial diversity that you find in a prairie is actually not a lot of available nitrogen. I'm really hung up on nitrogen more than anything else. So there are certain types of, I'll show you another graph, it may or may not be, it's another abstract graph. I was going to show you this no matter what. This is an abstract ordination of the bacterial communities out on the watersheds. When we extract the DNA, we sequence it, we get a list of 10,000 different bacteria that live there and all the relative abundances of all those different 10,000 different bacteria. That's too much information for anybody to assimilate. We put that data through a statistical analysis that shows us, there's a whole other, it shows us two samples, there's the difference in the species list between two samples. 2 points that are closer together on that graph have a more similar species list than 2 points that are further away. And there's, I know that's small, but there's lots more distracting stuff that will pop up here. The orange symbols were soil cores collected in the bison exclosure. The green symbols were soil samples collected outside of the bison area. The filled-in symbols were collected in 20-year burned watersheds, and the open symbols were soils collected in annually burned watersheds. So right here at this spot, I can see that the bison affect the bacterial diversity in the soil quite a bit. And kind of like the plant diversity, they have like a homogenizing effect almost relative to the fire. So you're more likely to see certain types of bacteria under the 20B and 20C than you are in in some of the annually burned watersheds. So I'm talking about this because I was talking about nitrogen. One of the main soil chemistry factors that varies out there with bison or any sort of large grazer is that these animals help recycle nitrogen themselves, maybe through help of their gut microbiome, but nitrogen availability is higher where you have grazers. And some of the species of bacteria that are the signature, like that characterize that difference, have a different role in the nitrogen cycle. They nitrify differently from some of the other types. So this is one of, and there's a lot of other things that vary there too. But this is one set of data that I'm really excited about. I have a different student this summer who's going to other grasslands around Kansas and Nebraska where they have bison to see if some of these indicators that we only know about from here, we've only really studied this here so far, is this actually characteristic of other grasslands in the Great Plains? And I know it's easier, or it's more, I have more confidence in saying there are certain species of microbe in the soil that are associated with different nitrogen cycling processes because we just know more about the nitrogen cycle, the specific species that are involved in the nitrogen cycle than we do about the carbon cycle. So when it comes to the carbon cycle, it's pretty wavy. So that's cool. This also, I started out by saying I can't necessarily tell you from one, if I just had one point with no context, I couldn't necessarily tell you where it came from. But I need these, reference points in order to draw the contrasts. And I also, we see this pattern out here on this landscape. We don't know yet whether it applies to other grasslands everywhere. So I still can't tell you that one type of bacteria is found in every single grassland everywhere.
The one infrequent fire bison outlier, is that?
I don't know. Yeah, that's a good question. There's always variation. What happened there? We definitely double checked it. There's another one. There's another one here. There's this one. There's one there. It's actually one of the cleaner graphs of this type that I've ever seen. Like one of the most clear contrast. I showed you the fungi and below ground class one earlier, which was just like they're shotgun graphs. There's statistically different separations. A few questions.
That's extremely interesting to me. I have a second question for you. You show difference between the bison, the grazed and the ungrazed. It's very obvious, but it looks to me like there's also, from the top to bottom, a difference between the frequency of fire.
Yes.
If you look at the shapes between top and bottom also.
Yes.
Do you agree with that?
Yes, and that's statistically supported. There is a different bacterial community in the annually burned ungrazed than the unburned ungrazed soils. That doesn't, first there's the fact of bison grazing, no effect of burn in the bison grazed areas, but outside of there, there's a big difference between the unburned and the burned. And that, this is one reason I can't tell you that we don't see that at the below ground plots, which also have trees versus grass. We only see that in this data set up there. There are some bacteria that live up there that don't live down here. And there's a hill slope effect, there's a soil depth effect. There's so many variables that go into determining some of these patterns. But up there, we collected those samples in a spatially explicit manner. So those dots weren't randomly collected. Where do you sample? It's a problem for all of us scientists. What is representative of a graph? We could go to one point, and what does that tell you about the rest of the landscape? So we sampled along these 10-meter transects, 2 samples from 10 centimeters apart, then half a meter, then a meter, then 5 meters, then 10 meters, and then extra transects. So we ended up with this log space axis, but I don't have a graph of in this slideshow for you. But the point is, the further away you get, the more different the microbial community is, no matter where you are out there. But just comparing among the unburned samples, that change was a lot steeper. So you could walk 100 meters, 100 yards out in 20B and find a very different microbial community, whereas here, like in 1B, you could walk 100 yards and find a much more similar microbial community. Same distance, more change in the types of bacteria you find in the soil. Another thing we're doing this summer is setting up an experiment to see if that's related to that distance dissimilarity, could be related to the openness of the landscape and the stuff that's literally blowing around and colonizing at distance. And then there's more trees and more litter on the ground that prevent different microbial cells from coming in. That's our hypothesis. We don't really know. So this question of movement of
Movement would have
cells,
something to do with animals.
very, very definitely related to the animals. And that's what we expected. We thought we'd see more homogenization where there was animal movement. But the point that you made about the difference on this axis between burned and unburned, we were not expecting that. And that could also be related to movement. Yeah, and it makes you think about, back in 100 years ago when bison were moving around everywhere, did they help the prairie have a more similar microbial community than it does now from point to point? We'll never be able to answer that question because we can't go back in time. Mike? This is one of my favorite graphs. Did you have a question?
Yeah, I had, so tell me if I'm right about this, there's no one correct recipe for microbes in a healthy prairie.
Some of the functional benefits that microbes bring to the prairie are shared among many different species of microbes.
Okay.
So there's not necessarily one perfect combination, as long as you get the ones that do the right jobs, like helping the plants get water and phosphorus, helping the manage the sands.
So, two, if you had, you said one way to get more microbes in the soil would be to have diversity of plants in the prairie. Would that lead to diversity of microbes in the soil, or the diversity of plants?
That's a general prediction that doesn't always hold, but it's often true. It's rarely not true. With more plant diversity, you get more microbial diversity. Yes, but not always, not necessarily.
And then a third part, if you were given a parcel of land that had been in heavy agriculture and it was given to you to turn into a prairie, what process would you follow to turn it into a prairie?
So we have, my husband and I have a tiny restoration of our front lawn out like halfway to Wamego. We've been working on for the past couple of years. So I can tell you what we did out there. But as I'm sure you're aware, I cannot tell you the secret to a perfect restoration. There's so many different ways of approaches to prairie restoration. Some might work at some sites better than others. For real, I mean, a lot of the hand waving I've done here is because the soil chemistry and the microbial community does vary from site to site based on the geology and then the land use history. So depending on the history of the site, you're going to have to just think about, your land better than anyone else. Think about what is most important for your own prairie restoration. So we have a relatively diverse lawn, but it's a lot of Bermuda grass, and then some other C3 grasses that we don't know. So the biggest challenge there is the competition between these existing lawn grasses and the prairie that we want to restore. So we do not put pesticides, they're not samples, but until that we put down prairie hay and mulch to add carbon because prairie plants are better at growing where there's less nitrogen relative to carbon than a lot of less desirable plants. So raising the C/N ratio of the organic matter in the soil can help the competitive dominance of some of your desirable prairie plants. And there are also soil microbes that are associated with that same characteristic of soil organic matter. So the propagules pressure, that's the probability of establishing you can afford to buy a lot of seed, that probably won't hurt. I mean, I'm not an expert in this. We were, I did sprinkle some soil that I may have had left over from experiments, like the jar experiments that we did. Again, thinking it wouldn't hurt, but I have no data on whether that.
Thinking you're adding microbes.
I think there's a lot of, yeah, exactly. But I don't know if that helped. I think what helped the most was being kind of patient. So the first year I was really depressed that our prairie one didn't turn right back into prairie. You probably know this better than me now. It's been 2 years.
So how do you
After
manage
2 and
that?
3 years, we actually got some big blue stones, some Indian grass growing.
How do you manage that for a long?
We might end up burning it. There's folks in the neighborhood who do, but we've been
The tall grasses
mowing it.
can't be mowed.
Yeah, we've been like cutting it down the side.
Your neighbors love you.
Because we don't burn. We might. It's so small right now. It's only been a couple of years.
You need a couple
I don't have
guys.
the secret. I know. Well, yeah.
There might be a couple
I'm
around
not.
here.
Those are too big for our scale. We're not that scale. I could get sheep, but I'm not sure. We're not gonna probably not gonna do that. But I appreciate, just opening, maintaining more niches for your plants to grow and whatever you can. That's always helpful. Go ahead.
Do you have any sites that are going to have livestock besides bison on them, or plans for sites to do some of this work on?
Yes. I wish there's so much more that there is a career's worth of work here, so that's good. We did collect some samples from the Patchburn Gray side along with these. They don't fit the exact same pattern, but we also just went up. It wasn't as thorough the sampling as we started asking whether the bison and fire affected and so on and so forth. I would like, to more expand in that direction. So the hypothesis about that difference is the effect of the bison on the microbes is related to the movement of the animals more than anything else. Cattle don't act the same as bison do. They don't necessarily range as easily across the landscape. But so the patterns may or may not be similar. I don't know. I won't know until I have more data. But yeah, they also, because of the way cattle systems, like the actual agricultural system works, cattle move longer distances than bison do nowadays. So definitely thought about that. But I can't answer it directly.
So the grazing effect, is that because of the cooking of the grass, or is that because of the manure, or a combination?
The question is, does the bison grazing effect, is it related to the bison actually eating the grass and opening up the canopy? Or is it related to the movement of the animals and maybe the deposition of microbes through the dung? And that is part of our dispersal experiment this summer. Is we'll sterilize dung and leave some unsterilized and see whether there's a fertilization effect or a dispersal effect.
I remember watching years ago our K-State greenhouses sterilize sterilizing the soil with steam. I was wondering if they were to bring, say, a container of sterilized soil were to be broadcast over the top in place of topsoil, how soon would that gain microbes and everything fairly quickly, or with it being sterilized?
Yeah, that's a good, the question is, how long does it take for a sterile soil to be colonized with microbes?
Yes,
In the native soil? I can't give you an exact answer. I haven't done that experiment. But it could happen pretty quickly. Days or weeks are what I'm thinking of as quickly. And it will depend on the physical connections through water or like mixing of the soils together. Yeah, it could happen pretty quick, especially if you mix it together a little bit. That question reminds me of a point about the inoculum for prairie restoration, and how kind of silly it was, but it couldn't hurt, but it was kind of silly of me to throw some soil left over from Konza on my front lawn, because there's a lot of soil already in my front lawn. After I'm throwing these tiny little particles of some other potential inoculum, the chance of the bacteria and fungi from that little bit of soil actually encountering the seed and the seedling of the plant that I want to grow isn't that great. So that dispersal idea is a challenge to plant inoculation with microbes. The best way to do it in experiments is the best way. I don't know what the best way is. You guys decide that. But I've seen scientific experiments that have shown the plant beneficial effects of certain bacteria very clearly. They show that they dip the plant roots right into the bacterial culture. And that's the only way you know for sure that your plant is associated with that bacterium. And that's how you run that experiment to show the plant beneficial effects of that bacterium. But out in the wild, in real life, unless you can grow seedlings and then dip them all in your inoculum before you plant them, it's pretty labor intensive. No way where your inoculum goes.
So you kind of got to the question I had in mind. Some of the early efforts Leopold and those guys did for restored prairies, they actually brought up chunks of prairie sod as an inoculum. Has anyone really experimented with that to see if it hastens the process?
Probably. So the question is, if you bring in actual sod as your prairie restoration, are you more likely to get recovery in the microbial community? I'm like extrapolating. But you're right, yeah, some of, the Curtis Prairie was restored using prairie sod from nearby. That's also labor intensive to cut up chunks of grass and bring it in. So I haven't seen any studies on that, but I can say, yes, we definitely have a better chance of restoring the soil chemistry and microbes if you're literally bringing some of those microbes in associated with the crab plants. I don't know how often is that really done anymore?
So, are you familiar with KU doing the research? I'm sorry, did I ever?
No.
Okay. KU's doing some research now using a tree spade that, they have a piece of prairie that's actually going to be built, a highway's going to be built on, so they're actually taking a tree spade, taking the soil out, going into a piece that won't go back to the other put it in, and they're trying to measure migration.
Good.
Okay, are you familiar with that research?
Yeah, has it been sites in Geneva?
I'm sorry?
What else? I might know the people who are doing it, but I don't.
I just don't know where they are with that. I just heard they were doing that.
It sounds vaguely, but I'm not sure exactly why. So the experiment that you're referring to is another kind of opportunistic activity where some native prairie is going to have a road built over it, and somehow some researchers at KU have gotten the resources to pick up a chunk of that prairie and move it into an old field and try to restore the stock and then actually track the recovery of the soil. That is an awesome experiment. A lot of the, like a lot of restoration activities, all of our science, we're usually resource limited for our science as well. So that sounds like somebody who's well prepared to take advantage of otherwise been a little bit like minor tragedy because we have other cases. Yeah.
This is a ridiculous question, but I have to ask. Do you have a favorite microbe?
It is the ammonia oxidizing archaea. Yeah, so I guess I was able to not that I don't like the others, but that's, yeah, and that's one group. But that's the group of, they're archaea, they're not bacteria, so they're more ancient and they're really divergent. That makes them interesting to begin with. But they nitrify just like the bacteria do. There's one group of archaea that does this really important job in the nitrogen cycle that we didn't know about until about a decade ago. And they actually are pretty abundant here. So that's the group that I'd like to work with. Thanks. Yeah.
So the work you've got going there, do you know if there's research that's been done similar on CRP lands, or are you going to look at CRP sites?
I do not know. So let's see. I can think of the second part of your question. Yes, of course. I'd be interested in expanding, the perspective on what makes a grassland management. This is an awesome site, but it takes a lot more work to see whether what we know here exactly predicts what's happening in the rest of the world. I can think of the first paper that comes to mind that's compared relic grasslands across the Great Plains was not they went to old township graveyards, which have never been plowed. They collected soils from those little graveyards across the Great Plains. So that's one of the only papers I know that has that breadth of.
I just thought they'd be great sites because they have known history.
Yeah.
And so you have that contractual management context and the history of the site, and we'll be able to piece that together with whatever we found. All that information isn't necessarily easily available, although I know it exists. Yeah, we can talk to somebody about it, for sure.
Yeah.
The important person is leaving in about a month, so I'll have that conversation soon and see if there's any interest.
Sure. Yeah, let me know. I mean, I know, I'm trying to remember, Greg Hausman down in Wichita, like there's a whole group of folks studying the plant and maybe the bird communities in a lot of the CRP lands.
Yeah, there's been a lot of work in other arenas, but I'm not familiar with any of the microbial sites. I'm just interested because the implications of what you showed and this other stuff and for their future contracts or management, it might be important.
Okay, one more question, then we're going to wrap it up. Right.
Have your studies included Fort Riley in with?
Not much, which is kind of, again, like we started here and trying to expand this time forward. We have the EPSCOR project that Jill mentioned. I actually work on the stream side of that, so we definitely were sampling on Fort Riley as part of our Kansas-wide stream biodiversity project. So yeah, I'd like to, again, limited by so many things, including time at this point, but they've got nice prairie, historic prairie, there's something about the history over there yeah, as far as like the next closest big tract prairie.
So we found out that there are so many more questions than we have answers to. It's a fascinating
That's
topic.
my life.
It's a fascinating topic, and we have lots of different research projects that we want Dr. Zeglin to go work on in her free time. But we want to thank you very much for